Hello, everybody, and also welcome from my side. My name is Christian, and I will be the chair for the first session of today, which will be called Population Genomics of Commensal Bacteria. We have two amazing speakers, so I would say we should jump right in. Just to remind you, if you'd like to ask a question, please use the Q&A section on the bottom of your Zoom window. You can also upvote questions to increase the probability of being asked in the panel discussion at the end of the session. And also don't forget to mention the name of the speaker you're directing the question to. So our first speaker for today will be Dr. Jesse Shapiro, and I'll ask Dr. Shapiro to start sharing his slides now. So Dr. Shapiro spent 7 years as a Canada researcher in microbial evolutionary genomics at Université de Montréal. He is now an associate professor in the Department of Microbiology and Immunology in the Genome Center at McGill University. Dr. Shapiro's lab uses genomics to understand the ecology and evolution of microbes, ranging from freshwater bacterial plankton to the human gut microbiome. So yeah, let's give a virtual applause to Dr. Shapiro.
Thank you very much, Christian and Sean, for the invitation. Very excited to be here virtually. All right, so my talk today is a question: does diversity beget diversity in the human gut microbiome? So this is a question that, well, not applied to the human gut microbiome, but at large, that is really an old question that we can probably trace back to Darwin or even earlier, looking at phylogenetic trees or just the mere diversity of life on Earth, and asking why are some environments or habitats more diverse than others? Why are some genera more speciose? So why are there so many species of beetle, for example, versus fewer species of apes, for example? So what are the controls of the rate of diversification in a genealogy like this? So broadly, we can break this down into abiotic factors. So things like temperature, pH, latitudinal gradient. So these are things that ecologists have been thinking about for quite some time now. For example, observing a latitudinal gradient of diversity, where there's more species diversity in the tropics than closer to the poles, for example. So maybe there's something about temperature, maybe there's something else about the tropics that increases rates of speciation, or in some other way allows more species diversity to accumulate. A little bit less well studied are biotic factors. So by biotic factors, I mean species-species interactions. So things that don't involve just chemicals or temperature, but actual competition within or between species, mutualism, predation, and I suppose the topic of the symposium is really about commensalism. So these kinds of symbiotic interactions and other ecological interactions. And there's broadly two competing models to explain how these biotic factors are expected to operate. These are not necessarily mutually exclusive, but you could think of them as two extremes, okay? So this is a cartoon plot where the x-axis is the standing diversity in the community. So you can think of it just as the number of species in the community. And how will that affect the diversification of one species in particular? Okay, so how does biological diversity affect the accumulation of additional diversity in the community? So the first model I'm going to call ecological controls. And this is basically a niche filling model where you can think of there's a limited number of niches. Each species occupies a niche. And so as we get to a high diversity in the community, those niches are just filled. And so there's no room for new species either to arrive by migration or to diversify, to speciate from a close relative. The alternative is what I'm going to refer to as diversity begets diversity, which is a sort of positive feedback in which actually increasing diversity in the community will, for example, open up more niches, new opportunities for species to either arrive or to speciate. And these are not these ideas date back to Elton in the 1930s and 40s, but this terminology comes from a nice review paper from Schluter and Pennell in 2017. So this ecological controls and diversity begets diversity. That's the recent terminology that I'm using. Okay, so what's the evidence for these competing models? So there's been some experimental evolution studies, mostly on Pseudomonas and other bacteria, and there's been a fair number of observations of plants and animals. And these have mixed results. So these are some experimental evolution studies where we find this example where niche occupation limits adaptive radiation. So this is supporting the ecological controls model. But we have counterexamples where we have things like high functional diversity in the community stimulates diversification in experimental microbial communities. So depending on the experimental setup, depending on the species used and the conditions, we do see contrasting results. Similarly, looking at birds, plants, fishes, we also see kind of contradictory results. Okay, so which one is it? Maybe it's a mixture of both, but actually the question I'm going to ask today is, bringing microbes out of the lab and kind of into the realm of plants and animals is just asking in nature, in the wild, how are bacteria responding to community diversity in terms of their potential to accumulate further diversity. So do bacteria in the wild tend to follow a DBD or an EC, ecological controls model? So to do this, this started from conversations with a collaborator, McGill at the University of Exeter, and a fantastic PhD student in my lab, Naima Mahdi. And to tackle this problem, we looked at different available data, but we eventually settled on this one, a communal catalog of Earth's multi-scale microbial diversity. So this is the flagship paper of the Earth Microbiome Consortium, which truly is a communal catalog of sampling bacterial diversity across many ecosystems on Earth. So this was appealing because there was standardized sample collection, and 16S ribosomal RNA gene amplicon sequencing that we could use. There are about 155,000 bacterial taxa in this data set that are defined as amplicon sequence variants, or ASVs. This is like an operational taxonomic unit that's maybe akin to species, definitely below the genus level. And just to get rid of sampling artifacts, we rarefied each sample to exactly 5,000 sequences in each of 2,000 samples. So we're trying to get rid of sampling effects, some samples being sequenced more deeply than others. So we try to have a very comparable data set here. And these are samples, as I said, that come from pretty much all over the world and from a variety of different biomes. So saline and non-saline aquatic environments, sediments, soils, host associated from both plant and animals, and this will come in later, but we're going to consider all of these. All right, so how are we going to distinguish these models? Okay, so as I said, we're basically plotting the initial diversity in the community. So for example, the number of genera, and then we're going to look at the diversity within a focal lineage, for example, a genus. All right, so if we think about a little phylogenetic tree here, this could be one sample. And in this case, we have one non-focal genus, this branch in black, and in purple is our focal genus that we're interested in, okay, which you see gives rise to 2 species or ASVs, okay? So we'll put a plot here where we have one non-focal genus and 2 ASVs per focal genus. Maybe in another sample, it could look something like this, where we have 1, 2, 3 non-focal genera, and then our focal genus gives rise to 4 ASVs. Okay, so we have a point here. We're accumulating these points. We draw a line through them. It's a positive slope. I'm going to refer to this as the diversity slope of this relationship. And this is consistent with the diversity begets diversity model because we see a positive slope. Conversely, if we see the opposite trend, this is consistent with ecological controls. which is the alternative. Okay, so we're considering this at the ASV to genus level. We can also consider a community assembly model where we're not really looking at it in terms of a tree, because especially at the resolution of 16S and in these particular microbiome samples, we might not expect diversification to really be happening. So we're not expecting to see the generation of this de novo diversity within these samples. Okay, so this is one model, but alternatively, we can consider a community assembly model, which the logic works pretty much the same, but the mechanism is different. So here, let's imagine that we start with one initial genus, and then it's able to accept two migrants from a non-focal genus in this community assembly model. Okay. Similarly, if under this DBD model, if we have 3 non-focal genera, it's able to accept 4 focal ASVs, okay, which you count them up and it produces the same slope. Similarly, under the ecological controls model, in this case, we start with 1 non-focal genus that gives room for 4 migrant ASVs to arrive, or if we start with 3, then that's room for 2. So you can see in this ecological controls model, there's basically room in the community for just 5 ASVs in total. Okay, so we can think of this in terms of community assembly as well. We can think of it on trees, and we can think of it not just at the ASV per genus level, but sort of deeper down into deeper phylogenetic relationships, like the number of genera per family, families per order, orders per class, and all the way down to classes per phylum. So looking at quite deep levels of taxonomy. Okay, so we go ahead and do this. We're going to do this using generalized linear mixed models, GLMMs, mostly to model this relationship of how the predictor variable, the number of non-focal genera, affects the response, which in this case is the number of ASVs per focal genus. And this work is now published in eLife, so you can go look at more of the details there. Okay, so the other nice thing about GLMMs is we can add a bunch of random effects that we think could also be affecting this relationship. So there's sort of nuisance terms potentially, like the sample ID. Maybe there's something weird about a particular sample, the submitting laboratory. So maybe there's some lab specific biases in sampling and sequencing, even though the EMP, does its best to make things homogeneous. And then there's some other effects that we think could actually be biological, like the type of environment or the genus, right? So maybe some genera have a tendency to accumulate more diversity than others. So we can account for these random effects in the model. And this is what the data look like. So this is a big slide, but just to give you a sense, this is looking just at the top 2 most prevalent taxa at different levels of the taxonomy. So going from phyla down to genera. All right, and again, we're just plotting this diversity slope. So community diversity on the x-axis, and then focal lineage diversity on the y-axis. And you can see that it varies across these different lineages. So the different lineages are the columns, so proteobacteria, Bacteroides, etc. And different environments are the rows. So proximal gut, the rhizosphere, non-saline sediment, and non-saline soil. So these are just some examples. And the red lines are significant positive associations, so supporting diversity begets diversity. And the blue lines are significant negative lines supporting ecological control models. And the gray are non-significant results. So you can see there's quite a lot of red, so quite a lot of support for DBD, but it certainly does vary quite a bit. Okay, so what can be explaining this? I'll get into in a moment. These results were using taxonomy, so this can be biased, right? The taxonomy is not necessarily great and not necessarily indicative of real genetic relationships. So we repeated this using nucleotide identity in the 16S genes to delimit sequence clusters independent of taxonomy. And we get broadly similar results. So this is combining, all environments, all taxa, so they're general results. But what you start to see here is more evident is the relationship is generally positive, but is certainly non-linear. So in gray is showing the linear fit. In blue is a cubic, and in red is a quadratic. Okay, so but just visually you can see that the relationship is positive, but maybe starts to hit a plateau. Okay, and these different panels are just showing equivalent from sort of like phylum level down to species genus level. So essentially different degrees of genetic relatedness. So yeah, we do start to see this positive relationship in general, but perhaps reaching a plateau. So another way of seeing this plateau is this plot where we're just plotting the diversity slope, okay, which as you can see is generally positive. So it's generally supportive of DBD, but how this varies according to the average community diversity in each one of these different biomes. Okay, so this is just looking at the phylum level, but we see it at finer taxonomic units as well. And what you can sort of see is that the more diverse the community, the more we kind of reach a plateau. So we get closer to a diversity slope of close to zero. Now the colors here, which you might have guessed, are in black. These tend to be host-associated microbiomes, and in red, these tend to be free-living microbiomes. So the free-living ones tend to be more diverse. Host-associated are more restricted in their diversity, and you can think of this as there's sort of more wiggle room. There's less niche filling, and there's sort of more potential for this DBD to, in a positive feedback loop, to increase diversity. But as soon as you get to kind of a plateau of diversity, you get into niche filling, and there's no more room for diversification. All right, so I'm not going to get into these details, but we did a bunch of null models. So randomizing the data in different permutations where we randomize the association between ASVs in a sample, and we don't observe these patterns. So we think this is something real and biological going on. I'm not going to describe all those controls, but they are in the paper that you can dig into if you want. All right, so can abiotic factors explain this pattern? Also something we dig into in the paper that I'm not going to get into. Short answer is no. There's some contribution of things like pH and temperature that do affect the diversity slope to some extent, but not as much as this variation with biotic factors. In particularly diverse biomes like soil, right, and sediments, there could be more of an effect of abiotic factors. And this is consistent with biomes that have reached a DBD plateau are kind of in a regime where their diversity is controlled more by abiotic factors. So you can think of biotic interactions important in the initial diversification, then it reaches a plateau and abiotic factors become more important. All right, so all of this is more on ecological timescales, right? So this is 16S RNA amplicon sequencing, limited genetic resolution, and we're really in sort of ecological community assembly mode where we're looking at between species interactions, changes in species frequency, this kind of community assembly. What about trying to get a little bit more towards that realm of evolution, where we're looking at changes in allele frequencies within species? Maybe we're actually seeing diversification due to de novo mutations or possibly strain dynamics, but certainly things are happening at a finer genetic scale than what is captured by 16S amplicons. So for the second half of the talk, I'm going to talk about some new results looking at data from the Human Microbiome Project, where we have shotgun metagenomic sequencing that lets us zoom in towards these evolutionary timescales. So we focus on the gut for a couple reasons. So animal guts have a strong DBD signal in the EMP. They're kind of up at the top corner there. And also for convenience, the HMP is another great resource. We have lots of data, 437 healthy human stool samples with over 20 million metagenomic reads per sample. So that's pretty useful. And this work is with Dr. Nandita Garut. So we teamed up to do this together. You'll hear from her later about some other research. But essentially what we did is we used a tool called MIDAS to process these metagenomes and first estimate within species diversity. So we define this as a polymorphism rate as the proportion of single nucleotide variants or SNVs between a 0.2 and 0.8 frequency. So intermediate level frequency polymorphism. This is kind of like pi in population genetics, if you're familiar with that. And we have some coverage and other filters for quality that I won't get into all the details. And we're also going to estimate community diversity also from MIDAS, either as richness, so just the number of species, or the Shannon diversity, which tells us a combination of richness, but also the evenness, so the relative abundances of those species. All right, and so the rest of the work I'm going to talk about is, as I said, with Nandita Garut's lab. So both Naima, a PhD student in my lab, and also Daisy Chen, who is a student with Nandita. And so in this first plot, we're just looking at 68 species in the gut microbiome that are present in at least 4 samples. And you can see, so plotting these 2 measures, Shannon or richness, 2 measures of community diversity against the polymorphism rate within a focal species. Okay, so different colors are different species. I'm not going to show you what each of them are, but you can see that these slopes are generally positive, also generally supporting DBD. Here's what the species are. Okay, so there's some familiar gut species. You can see in bright green, Bacteroides vulgatus that I'll come back to is pretty visible as showing a positive slope. All right, a couple things you'll notice in these plots is the relationships are non-linear. Okay, so we're drawing straight lines through them, but if you look at it, it's probably not super linear. Second thing is that the polymorphism rate varies quite widely. So, closer down to here, that's consistent with de novo mutations, but way up here, it's really a lot of mutations. So that might actually be tracking strain turnover, not de novo mutation. So I'm gonna get into each of these 2 observations in a bit more detail. So first, we can account for this non-linearity by fitting generalized additive models that don't force linearity. They can fit a smooth curve. And so you can see these are what the smooth curves look like for both Shannon and richness. Here we're including sample coverage as a covariate to control for those sampling effects. And you can see that the trends are positive and we do see this sort of plateau. And this is even more apparent. Another way of doing this, instead of including coverage as a covariate, is to just work on rarefied data like we did on the EMP, and you really do see this plateau. All right, so this range of polymorphism, the second point, we can explicitly get at this. Okay, so high levels of polymorphism could be due to strain variation, not de novo mutation. So to do this, Ricky, another one of Nandita's fabulous students, used a tool called Strain Finder to estimate the optimal number of strains per species. That turns out to vary between 1 and 4. And you can see that there's some interesting results here where using Shannon diversity as a predictor of strain count, the results are generally positive. So most species have a positive correlation, but we actually see the opposite effect where most species show a negative correlation with richness. So these 2 measures of diversity are telling us different things, which is not necessarily unexpected, but it's sort of an interesting observation. And we get similar results. So positive strain diversity relationships for Shannon, but negative for richness at all these different taxonomic levels. So it seems that much of this DBD trend in the Human Microbiome Project can be explained by strain dynamics, not just de novo mutation. And Shannon and richness behave differently, even though we do expect them to be correlated, but imperfectly. All right, so the last question I'm going to ask is, okay, how do these dynamics really play out over time? Okay, so far we've really just been looking at snapshot samples, but can we actually see this happening in real time? So luckily in the HMP, there are a number of people that are sampled twice, between 6 and 12 months apart. So we can look at not just SNVs, but SNV changes between time 1 and time 2. So we're defining these as SNVs that are undetected at the first time point, so below a certain frequency cutoff, that then are detected at the second time point, or vice versa. Okay, so an SNV change. And gene content changes as well. So we can look at gene gains that go from a copy number that we consider undetectable, so effectively 0 at time t, that go to a copy number of about 1 within a range of confidence at time t plus 1. Or losses are vice versa. Okay, so going from present to absent. And we can again look at the different predictors, so the community Shannon diversity or community richness as a predictor of all of these different responses at the next time point, okay, time t plus 1. And these are the results. So these are GLMMs again, and you can see that there is a significant positive effect of community diversity on SNV changes, okay, consistent with DBD. But sort of unexpectedly, also a positive effect on gene loss. So more community diversity at one time point is associated with a tendency for genes to be lost at the next time point, and no significant effects on gene gains. So to think about this in a bit more detail, we looked at 2 individual people densely sampled over time to see if we can replicate these results. We're looking at B. vulgatus, which happens to have one of the highest DBD signals, is also very prevalent in these individuals, and has 3 dominant persistent strains. So these are results published in these 2 papers by Zhao and Lieberman et al. You'll be hearing from Tammy later, and you'll also be hearing from Mathieu later as well. So this is some of their fantastic work that we piggybacked on. All right, so here is the time series from one individual. Okay, so for B. vulgatus, the SNV count. Okay, so again, sorry, this is actually SNV changes. I should relabel that over time, mapped against species richness in the overall microbiome over time. You can see that they both have like a little bit of a bump here, which results in them being positively correlated. Same thing for looking at species richness. We also see this positive relationship. Now, just looking at the scale here, these are thousands of SNVs. So again, this suggests that these dynamics are actually driven by strain abundances rather than de novo mutations, which is consistent with we know there are different strains of B. vulgatus. There's no significant trend of gene gain being affected by community diversity, but we do see this positive trend with gene loss, which is what we saw in the HMP. Again, these are hundreds of gene loss events, which are probably more consistent with strains with fewer genes replacing strains with more genes in correlation with community diversity. So these are probably not independent events. This diversity slope kind of goes away with longer time lags. It's strongest, at close time lags up to about 48 days. Similar with this gene loss effect, although it's a bit more stable. So we see the positive relationship between Shannon diversity and gene loss persisting for longer. Why could this be? So why does higher community diversity correlate with gene loss? Well, something that could explain it is the Black Queen hypothesis from Morris, Lenski, and Zinzer, which basically posits that genes with leaky functions, so that produce metabolites that go out into the community, these can be lost by a focal genome if they're encoded, if they're complemented by another member of the community. So in an extreme example, an ecosystem of just one species, well, that species has to encode all the essential gene functions, but in a more diverse ecosystem, a focal species can lose certain genes as long as those functions are provided by others in the community. So do we also see this in the HMP? So back to the HMP. So we're going to plot the number of genes in a sample in the community versus the number of genes in a focal species. We see a positive relationship. So this does not really support the black queen, but it's also kind of expected given that we see a positive DVD signal and we expect community gene diversity is expected to correlate with community species diversity, which we know show a positive relationship. However, if we look at pathways instead of genes, so metabolic pathways, we see a really strong negative correlation, which does seem to support a black queen model at the pathway level. This is just what the raw data look like. So those were fitting GAMs. You can see that there's, kind of a range of pathway diversity in red. These points are E. coli. So we thought, oh, is there an E. coli effect because it's just really well annotated? So maybe there's some outliers, but even in down here where the pathway diversity is not so large, we do see generally negative slopes. And these are a few species in different colors that do show a slight positive slope. So there is some variation. All right, so just to wrap up, there is a huge value of these community data sets, HMP, EMP, the Broad Open Biome data set with those B. vulgatus data sets. So huge value of reusing these data sets. I'm going to call it data commensalism. I know data parasitism was thrown out as a term, but this is a commensalism session, so I'm going to go with commensalism. And this complements early work from experimental evolution and in plants and animals. So to summarize, we see both DBD, so both positive and negative black queen type biotic feedbacks, and these are likely important in shaping diversity in the gut. The mechanistic nature of these feedbacks is unclear. So I've showed you just broad correlations. So actually digging into what's going on, are these cross feedings, are these niche construction interactions? This will require more experimental work. So I will leave you with that. I've already thanked most of the people who did the work here. There's a lot more faces than there are funders because this is sort of fundamental research that we did without a lot of funding. So I'm very grateful for basic research funding from NSERC and the Canada Research Chairs Program. So thank you very much, and I'll stop there and look forward to the discussion after.